Hi, my name is Josephine Byers, and for my presentation today, I'll be demonstrating my research on curing beta-thalassemia. This is my gene therapy proposal for the research grant committee at Byers University. To quickly cover what I'm going to be discussing today, I'm going to kind of break down the molecular level of how it works. I'm going to be talking about the market competition and other treatments for the disease. I'm going to be discussing quantification and validation techniques, also the safety and the efficacy, and the ethical guidelines of my therapy. Now, just to quickly discuss what beta thalassemia major is specifically, major means that it's the more severe form of the disease. But either way, beta thalassemia causes missing or defective beta globin genes, which disrupts the function and the structure of the hemoglobin, which is the main component of our red blood cells. So this causes them to be malformed, kind of similar to sickle cell disease, which can cause fatigue, blood pressure changes, things like that, and often results in organ failure and death. So most people with the disease don't tend to live very long. Now, this disease specifically is caused by a substitution in the DNA, which means one nitrogenous base was flipped, and that disrupts the entire function and production of hemoglobin altogether. Now, how is this Cas9, CRISPR-Cas9 actually going to work? We use engineered nuclease to cut DNA at a specific spot using a double-strand break. You could also, instead of cutting and going in, you could just supply the correct DNA sequence and introduce that new segment. There's different ways you could kind of go about it, but there's also 3 different engineered nuclease you could use, but of course the most common for CRISPR-Cas9 is the Cas9 protein. There are 3 big components that we need for doing this. We need the actual Cas9 protein, we need the guide RNA, which is a sequence of 20 nitrogenous bases involving the PAM site, and we need, of course, the PAM site. So the guide RNA is what tells Cas9 where to bind in the genome. That's why it's 20 bases long. The problem with this is that there is a very commonly off-target cutting, which means it cuts at a different spot in the genome that has the exact same complementary base pairs. So often when we're designing our guides, we need to pick one that isn't too prevalent in the genome. The PAM site is what actually allows Cas9 to bind. That's NGG. N could be any of the nitrogenous bases. And the Cas9 protein is what actually does the cutting into the DNA to change it. Now, just to kind of give a little visual diagram of this, you can see the PAM site there in red, the RNA opening it up, and the actual Cas9 doing the cutting to change the DNA. Now, there are quite a few current treatments for this disease, but often they can end up in iron buildup, and they can also have negative effects. None of these treatments are curative, whereas my treatment would be, and they just kind of provide a short-term fix. It's something you need to keep coming back for. None of them are completely going to get rid of it, and they also have a lot of negative side effects. If my therapy can be tested and proven to work efficiently, then you wouldn't have to keep coming back in for these treatments. It'd be kind of a one and done. Now, to actually validate that my treatment is working, we would use in vitro cellular studies. Now, the 2 biggest things we want to make sure we're validating is if it can get into the nucleus and if it can actually make the cut. So you have to test for delivery efficiency of the RNP and the cutting efficiency. Those are the biggest 2 things we need to make sure we're validating. Now, to do this, one of the most common techniques is electroporation. That's where we can actually kind of shock the cell, creating small pores to actually make sure it's getting into the cell, which is a very cool technique we can do in the lab to test for that. And we can also quantify it. Now, once the genetic engineering is supposed to be done, we can PCR the DNA, amplify it, and then we can use 2 different kinds of sequencing to actually sequence the DNA and see what different bases we have. This will produce sequencing reads, and we can compare the reads to see how many of the mutant DNA strands we have and how many are actually healthy. So how many DNA strands actually were changed and altered, And typically, we've seen about 40 to 60% of DNA being altered, which is enough to make a significant impact on the client. But these are just different ways we can actually quantify and get a value for how much of our DNA is changing. Now, preclinically, there's a lot of different safety and efficacy testing that we need to do. First, you want to test on the cellular level, so testing the DNA, the mRNA, and the different protein. As I said before, we can check the percentage of the DNA that has the mutated gene and how much of it actually has the healthy gene. Similarly, we can check the percentage of the corrected mRNA production. These typically go hand in hand. And then we can also check on the protein level, the hemoglobin level and profile, and if it's functioning correctly from that beta globin gene. Now, following that, we can move on to animal testing. Typically, we use mouse models and we use non-human primate models. For the mouse, we can use immunodeficient mouse, which have a weaker immune system, or we could use a mouse model with the actual disease. This provides a more human-like situation before we actually put it into a human. Specifically, that non-human primate model has really high similarity with humans, so you can really see how the treatment would react in a body similar to ours. But once that's all safe, we can actually move on to the clinical trials. First, we need the FDA to approve it, giving us an IND. And all of our patients and people involved in the study need to make sure they have informed consent. But once those two barriers are crossed, we have phase 1 trials, which is just for safety, first time putting in a human, making sure is it safe to be putting in. Then we move on to the phase 2 and phase 3 trials, which are actually testing for efficacy and if it's doing what it's supposed to, and also further safety. And then phase 4 trials is when we can actually start getting data and information about deeper things into what's happening within the body. Now, when we move on to ethics, there are 4 different ethical things we can take into consideration here. First off, the treatment that I'm proposing will definitely maximize benefits to patients. It will reduce all of their negative symptoms, including organ failure and death. So this is definitely something that will benefit them and will help them. It also minimizes harm. As I said, you're getting rid of all of those negative side effects. You're not inflicting unnecessary pain upon the body. You're doing something that will actually help them. And they have autonomy. They can choose whether they want to participate in clinical trials, and they can choose if they would like to undergo the treatment. So it's not being forced upon anyone who doesn't feel safe and willing to take the risk involved with most treatments as such. Now, the one thing is justice. As of right now, it's probably not equally available all over the world because it can be really expensive. But in this case specifically, we would be curing a life-threatening disease, which kind of overrides that a little bit because saving some lives is better than saving none. But with more testing and with more participants, if we can further advance this therapy, then it will start to probably become more accessible and able to reach these different areas. And with that being said, thank you so much for listening to my presentation. I enjoyed all of this program and I learned a lot from it. So thank you very much. It's probably going to take me a second to figure out how to turn this thing off. But I hope you have a great rest of your summer and thank you so much.